for putting together this meeting. This is my first time in Brazil. It's been fun so far, and I'm looking forward to the weekend too. Is this on? Is it working now? Okay, excellent. All right. So what I would like to tell you about is work that we've been doing over about the last 7 years in my lab in Alberta on trying to understand what's happening in protein misfolding at the level of single molecules. Now, you know, this audience, I don't really have to explain the importance of structure for function, right? So the point is that if something goes wrong in the folding, and we end up with the incorrect structure, then we're not going to have the right function. And as it turns out, there's, in fact, many diseases that are associated in one way or another with protein misfolding, from a whole host of neurodegenerative diseases to things like diabetes, so metabolic diseases, connective tissue diseases, etc. Our particular interest is actually in the neurodegenerative diseases, and one of the common clinical features of many of these diseases is that you get these deposits of fibrillar forms of misfolded protein, whether we're talking about prion diseases or Alzheimer's or Parkinson's or many others. Now, the regular folding protein problem, you know, telling us how we go from an unfolded protein right after synthesis in the ribosome to our native structure, this is something that we've been studying for over 50 years collectively, and there's still surprises that are getting thrown up by this, as this is not exactly a simple problem. But misfolding is unfortunately quite a bit worse because it's incredibly heterogeneous. There's a whole lot of different things that are going on. And this makes it very difficult to understand what are the important events in here. But it's also an opportunity for us as single molecule biophysicists because, as it turns out, single molecule methods are actually very well suited to trying to piece together what's going on in a complex situation like this, where by avoiding ensemble averaging, we can do things like directly observe very rare or transient events that might play an important role, like, for example, rare things that nucleate misfolding for the first time. Or if we have a heterogeneous mixture like we do in the misfolding problem, we can basically pull out each of the different subpopulations and characterize them separately. And in fact, we can follow a single molecule as it changes between these many different possible structures, and even, in fact, follow what's going on on the transition paths in between these to learn about mechanisms. Now, I mentioned that we're doing this with force spectroscopy, so why are we using force? It brings a number of advantages. First of all, we can achieve quite exceptional resolution these days and sensitivity. So you can get spatial resolution as good as an angstrom. That was achieved a bit over a decade ago. Most recently, we've gotten down to the microsecond timescale for time resolution, both with optical tweezers and with AFM. And you can get exceptional sensitivity to rare states, as low as 0.001%. The other thing is that, of course, a force integrated over distance gives us an energy, and so this allows us to manipulate the energy landscapes in a very controllable way. And in fact, it turns out there's multiple different ways to reconstruct landscapes quantitatively from these kinds of measurements. We heard some about that yesterday from Olga, and I guess I'll be talking a bit more about that on Monday. And finally, something that's particularly important for misfolding is that force changes the structure without having to apply these global changes to the conditions of the experiment, like changing the temperature or changing the pH or adding a denaturant, all of which actually have important effects on the aggregation process. So this allows us to separate those things out, and then, in fact, we can go and test what these kinds of changes are doing separately. Okay, so that's the motivation for using force. The particular approach we take is using optical tweezers, and this gives you the idea of the experiment. We take the molecule we're interested in looking at, we attach it to beads, which are held in optical traps, and then we can move these traps apart to apply tension. And as you raise the amount of force that you're applying to this molecule, at some point pieces of it are going to come apart, unfold, and when they do unfold, because of this tension, they get stretched out. So you see this change in the extension. And this extension then becomes our reaction coordinate for following the progress of the folding. And it basically reports directly on the instantaneous structure, so how much of the protein has been unfolded. All right, so what I'd like to do today is talk about two different systems that we've been looking at. The mammalian prion protein PrP, which we've been studying for about 7 years now, and then some more recent work that's as yet unpublished on superoxide dismutase. So let's start with PrP. And this protein, I guess, burst onto the public consciousness a couple decades ago when, I guess it's now almost 3 decades ago, when mad cow disease was discovered. And so this was first discovered in the mid-'80s in the UK. And it's this fatal and infectious and progressive degenerative disease. It's got a number of distinct symptoms. These include extreme sensitivity to touch and sound, loss of balance, actually aggressive behavior is one of them, and then eventual death. And this gives you a sense of how BSE progressed in the UK. This is the number of confirmed diagnosed cases. So there are over 185,000 so far, peaking about 25 years ago in the mid-90s. Now, North America has had 24 cases. You'd think, oh, that's not a big problem, but it turns out that the very first case of homegrown BSE cost something like $7 billion because the world closed its doors to the meat market, right? And North America actually produces a lot of beef. Brazil had its first case in 2012, and they encountered the same problem that a lot of places in the world closed their markets to exports. So it's actually it's still an ongoing problem. 2012 is quite recent. You can see, you would think from this that it's basically gone away, but it's actually still a problem. All right, and of course the issue here is that the meat from the infected animal is capable of inducing a version of the human prion disease. It's called new variant Creutzfeldt-Jakob disease. And in fact, BSE is only one of a whole family of diseases that affect many different species. They all have the same symptoms of, you get these amyloid deposits of PrP, and then you get these vacuoles in the brain. So that's why it's called spongiform encephalopathy, because that is spongy brain tissue. And this disease is 100% fatal. There's no cure for it, and everyone who gets it, and any organism that gets it, dies. In all cases, we know now conclusively that it's caused by the protein PrP. So this is a highly conserved membrane-anchored protein. It's alpha helical in its native state, and somehow it can turn into this infectious form that's beta-rich. And it's infectious in the sense that when this one of these comes into contact with natively folded protein, it converts it into more of the infectious form. Now, the interesting thing is there's been 2 Nobel Prizes given out for prion diseases already, and yet there are many really important questions that are still unsolved. So, for example, we don't actually know what this protein is doing, and why is it so highly conserved. It's clearly apparently important. However, if you delete it, you can make mice that have no PrP gene, they live, and they're completely immune to the disease. They're sickly, so it's not, obviously it's doing something, but some other proteins must be able to recapitulate its function to some extent. But there's many suggestions, but it's still not understood really what it does. A bigger one is that we do not know still what the structure of this infectious form is. People have been going at this for decades. We think we're getting close with this recent work on fiber diffraction, but still not there yet. We don't know how the misfolded structure is formed in the first place, or how it propagates. We know it's some form of seeded polymerization, but there's a lot of debate over exactly what's going on. And as it turns out, we don't really know how this infectious form of it actually kills off neurons. Now, we are focused on this question here. And so we are looking at this using optical tweezers. So this is how we do it. We take, we're studying hamster PrP. So hamsters are a nice model to use because they're actually quite susceptible to the disease. Not all species are equally susceptible. And we use a fragment of it that corresponds to the fragment of the infectious material that's protease resistant. So we know it's really stable in the misfolded form. Then we add cysteines onto the end so that we can make disulfide bonds to DNA handles, and those handles are then attached to the beads. And we do all these measurements at neutral pH, in near physiological ionic strength, at room temperature, and then without chemical denatures. So what do we see when we look at this? Well, one of the measurements we can do is just move the beads apart at a constant speed to ramp up the force, and plot the force as a function of extension. So we first stretch out the handles, and then this is the interesting part where the protein unfolds. You get a sudden length change, and then we keep on stretching everything out. We can bring it back, and it retraces its steps, but there's a little bit of hysteresis because we're actually changing the force fast enough that it's slightly out of equilibrium. Of course, we can do this lots of times. So here's 100 pulls on the same molecule, all overlaid on each other, and you can see that they line up very nicely for the folded and unfolded states, but there's a certain amount of randomness in the actual force, and Olga already talked about the fact that this contains a lot of information about the landscape. Here we can do something kind of important for us here when we're interested in misfolding, is to check what is the length change. So we can fit these force extension curves to polymer model like worm-like chain. Here we've got a worm-like chain just for the DNA handles. Here's one for the handles plus the unfolded protein, so we can measure the length change. What we find is that it's 34.3 nanometers, averaged over about 4,000 pulls, and that matches just about exactly with what you would predict from the high-resolution structure of this particular form of the protein. Okay, now, these kinds of measurements are a little bit tedious, ramping the force back and forth and back and forth and back and forth. So instead, if you've got a situation like this where it can coexist in two states, you can just pull it out to one of these forces and then sit there at constant force and watch it hop back and forth. So this is one of these constant force measurements where it's just bouncing back and forth in equilibrium now, so it's not non-equilibrium like the previous measurement, between a high extension force, which is corresponding to the unfolded state, and a low extension force that corresponds to the folded state. In fact, if you measure that distance, it's the distance you expect. And then, of course, we can change how much unfolded versus folded we have by changing how much force. At higher forces, it's more unfolded, and at lower forces, it's more folded. All right, so these are the kinds of measurements we can do. Let's now see what we can learn. So the first question we wanted to look at was whether we could find this partially unfolded intermediate that had been suggested quite a long time ago, over 20 years ago, to play an important role in conversion. The idea is basically that this partially folded state here ends up being the right place to branch off and go into a different structure. And over the years, a variety of different hypotheses have been made for what this might be, but the evidence has been fairly conflicting and generally indirect. And so this is one of the advantages of single molecule approaches, is it's actually very sensitive to intermediates, so we can look for these directly. And the problem is that when we do this, we see, basically nothing in the middle there. Nothing obvious, at any rate. And just to give you a sense of what an intermediate looks like, here's a trace from a DNA hairpin that we engineered the sequence. We made blocks of ATs and GC base pairs so that there is an intermediate right here. And as you can see, if we just blow up one of these transitions, you get this intermediate state occurs on every transition. There's a little blip there, and also as transient excursions out of both the folded and the unfolded state. So that's what we ought to see, but we're not seeing that. And we went through a whole bunch of, of course, it's hard to prove the non-existence of something. Really, all we can do is put an upper bound on how long it must last for and how often it must occur. But after some, extensive statistical analysis, we find that the lifetime can't be any longer than about 50 microseconds, and the occupancy has to be less than about 0.001%. So if it's there, it's probably not important, is our conclusion. However, while searching for this on-pathway intermediate, we found evidence for actually a lot of off-pathway intermediates. And so my claim is, if you look at this trajectory here, so that's the unfolded state, this is the folded state, and then you see a whole lot of spikes coming down from the unfolded state. And my claim is that those are intermediate states. It's a little bit more convincing maybe if we blow it up. So now my claim would be that these two levels are the misfolded states. It's still not hugely convincing because it's roughly on the same time scale as our Brownian motion noise, you can see, of the beads. However, it's more convincing if you take just look at the distribution of extension values in this state here, and you fit this to our point spread function for the trap, which is where we take just a DNA handle, no protein, and measure what distribution of positions we get. And we get this parabolic shape here in our, I mean, it's actually Gaussian, but I'm showing it on a log plot, so it looks parabolic. And there is a distinct residual there. And in fact, we can fit this residual to the point spread function, and we need 2 point spread functions to fit it. So we see 2 different misfolded states. They're just quite rare, less than 1% of the time. And in fact, my students spent a long time looking through this data, and about once every 10 minutes or so, you see something that lasts about 50 microseconds. So it's longer lived, but it happens much rarer. The point is, overall, that we actually have frequent, but very short, generally short-lived attempts to fold into some alternate non-native structure. And that never leads anywhere. It's sort of a futile state. So this allows us to put together this pathway for what the monomer of PrP can do. We see no on-pathway intermediates, but out of the unfolded state, we see these 3 different misfolded states. We can estimate their lengths based on the length of the position of the state on the reaction coordinate. And the key point here is that it seems like the unfolded state is most important for misfolding. Curiously, these misfolding rates are actually higher than the native folding rate. And also important, none of these misfolded states is actually stable in the context of a monomer. So they're all unstable, and you always end up back in the natively folded state. Now, this is all monomers, and as we've seen here, we don't find any stable misfolded states in a monomer. And in fact, this is corroborating a suspicion that it existed for quite some time in the field, that the misfolded state is only really stable in the context of a oligomer. So our next step was to look at aggregates, so small oligomers, and of course the smallest oligomer you can look at is a dimer. So being physicists, we went from a monomer to a dimer to build up what's going on. And so we basically took 2 monomers and stuck them together to make a tandem dimer, like you often do in these kinds of measurements. Now, there's actually some reason to believe this may be biologically relevant, because dimers are actually toxic both to cultured cells and to animals. And in fact, you can, some people have raised antibodies against dimeric structures, and they have some minor protective effect. So, you know, they might have something to do with what's actually going on in the disease. So our picture has changed now from this to this, where we just make this tandem dimer. They're stuck together end to end. And just to remind you, this is what the pulling curves of a monomer look like. So if we had, in fact, this picture that I've showed here, where I've shown independently folded monomer domains, you'd expect them to rupture independently, and you'd basically get 2 basically equal rips in your curve. And this is seen for, in hundreds of papers looking at tandem repeat proteins. But what we see is something quite different. So it's not 2 monomers, clearly. And instead, it's converted to some kind of stable misfolded form. Now, when we do this repeatedly, we actually see 2 different types of behavior coded in red and black, but we never see in like 2,000 pulls native folding. So these 2 populations, very creatively, we called them type A and type B. So type A has this large rip there. Type B has a whole lot of subtle things going on. So we can do the same kind of analysis I did with the monomer, where we identify the transitions. In this case, we can see 3 transitions, distinct transitions, meaning that there's at least 4 states. And then we can fit the length changes by fitting it to the worm-like chain model. So here, for example, the total length change upon unfolding is 81 nanometers. Now you might be curious, how do I stick 2 things that are 34 nanometers together and get 81? And the point is that actually it turns out that the N-terminus of PrP is disordered. So it doesn't contribute to this length. But now when we put it into a dimer, it's apparently contributing to the structure of the dimer. This little thing up here actually occurs in both types. It's the same length, it's 16 nanometers, so it's a little bit less than half of the normal length of the normal structure. And then this guy here has a total unfolding length of 53, so it's about 1.5 times the monomer size. But actually, this isn't all that's going on. You can see that there's part of the curve that sort of slides from one worm-like chain to another. And so this does not have a nice, sharp transition like I've been showing you up until now. And the way to understand this is that you get these sharp transitions when your sampling rate is fast compared to the lifetimes of the states you're looking at. Okay, because then you get a nice discrete transition. If the sampling rate is actually slow compared to the, so you're actually going back and forth quite rapidly on the time scale that you're making this measurement, then what you end up with is basically an average over the, and you get a much smoother curve. So that's actually what's going on here. And we can take basically average all the force extension curves to get a smoother curve there, and then fit this to the a model of the average force extension curve you'd expect, where you have two parameters, the unfolding force, the average unfolding force, and the length. Now, if we try to do this with just these states 2, 4, and u, what we find is that we can't actually get the middle to fit here. We get these small but systematic deviations. However, if we allow for one more state, it actually fits really nicely. And then we can do the same thing for, okay, so that tells us that we have this pathway 2, 3, 4 to unfolded. We can do the same thing for type A, because again, you see something that sort of slides into this state here. And again, when you add an extra state, you get rid of that systematic residual, and in fact, the numbers for this state ends up being the same as up there. So it's all self-consistent. So this suggests that for type A, what we're seeing is this pathway 1, 3, 4U, where we skip over 2. All right, so that's unfolding. What about refolding? It turns out that all the refolding curves match the second type of unfolding. So that's our type B unfolding, and that's the refolding. So that's telling us that the refolding pathway goes backwards, U432, which leaves you wondering, where does state 1 come from? And in fact, so we puzzled about this for a while, and we thought, well, maybe it's slowly forming at 0 force, and so this is why we don't see it. And in fact, if you change the amount of time you wait at 0 force between pulls, then you increase that amount of time, you get more of that state forming. So it's just a slow process, and it's happening at fairly low force. So we're not seeing it. We actually have like 1 or 2 curves where you can see it directly when we looked closely at the data, but most of the time you just don't see it. So this gives us this picture where for the complete pathway, where these 5 states, and this process here is slow, so we either see it sort of jump from here to 3, 2 like that, or go along the top. Now, at this point, we can start using the formalism that Olga was presenting the other day, and look at the energy landscape properties by examining the distribution of forces that we get from this. So this is the idea shown here. This is just for a 2-state system, not a multi-state system. But if we have a very stable structure, it's got a high barrier, so 0 force is not going to unfold. But as we tilt the landscape from applying force, at some point the barrier becomes small enough that you'll start seeing unfolding. As you keep on tilting, you get more and more. And so you get this expression here that describes the shape of that distribution in terms of these 3 landscape parameters: the distance to the barrier, the height of the barrier, and then the rate for going over it. So we basically take these curves, we turn it into a histogram, we fit it to the theory, fits very nicely, then we do this for thousands of curves, and we can get these 3 parameters for, this is for the monomer, and of course we can do the same thing for the dimer. We treat each of the transitions independently. Now, Olga has in fact shown that normally you're not allowed to do this because there are correlations between the forces. But it turns out that in this case, the transitions are quasi-equilibrium, and the forces are uncorrelated. So we can treat each of them as 2-state systems. And then we can put together a picture of the landscape for the whole thing that looks like this. We can measure the total distance and energy from the unfolded state to the folded state here and compare that to the monomer. And in fact, we find that it's 2 kcals per mole more stable than 2 independent monomers. So this misfolded state is in fact the thermodynamic minimum for the dimer. Now there's something else that's quite interesting that we can get at from this analysis, and that is that we can actually get at the underlying diffusion coefficient, right? And the diffusion coefficient is what connects the landscape to the kinetics. So we're using the Kramer's theory, which is also the basis for Olga's analysis, right, which relates the rate to the, so you've got your Arrhenius expression here, and what we're interested in is this diffusion coefficient there. So if we know the height of that barrier and the curvatures, which we get from basically the other parameter, which is delta x double dagger, that ends up together with the height that ends up giving you the curvature. Then we can actually pull and the rate, and that was the third parameter from the fits. Then we can pull out D. And of course, D is interesting because it tells us about internal friction in the protein, and it's related to the speed limit. And so here's the expression that relates those three parameters from Olga's theory to the diffusion coefficient. And when we apply this, what we find is that, and we also did this to the native structure in the monomer, we find that we get actually a very slow diffusion. So it's 1,000 nanometers squared per second versus 1,000,000 nanometers squared per second for the monomer folding natively. So it's 1,000 times slower than for native folding. And remember, this is for the exact same protein. So if we compare the native and native folding and misfolding, so this is the landscape we got from actually a slightly different method developed by Gerhard Hummer and Tilla Sabo, but same, it's completely equivalent. The answers agree very quantitatively. So here's the two landscapes. What we're saying is that even though I've shown this as smooth, it's actually quite a bit rougher because of these, presumably because of non-native interactions that are generating all sorts of localized traps that are slowing down the diffusion. And we can use Svansig's theory to estimate the extent of the roughness, and we get about 2 to 3 kT. And this is interesting because there's been this long-standing presumption that misfolding landscapes are much rougher than folding landscapes because there will have been no evolutionary pressure to, selection for good behavior on a misfolding landscape, right? But this now validates that quantitatively. Okay, last story on the prions that I want to talk about. As you know, our cells have these molecular chaperones that help to reduce protein misfolding naturally. And in fact, many people view these misfolding diseases as arising from failures in the proteostasis machinery, including the chaperones. So for a long time now, one of the strategies for developing therapeutics has been to say, well, maybe we can find some small molecule that actually helps the chaperones. So they basically act as so-called pharmacological chaperones. And a number of candidates have explored for actually many different diseases. Here's just a few of many proposals for prions. Now, one of the problems with this is that in none of these cases do we actually know what these molecules are doing to the misfolding, because misfolding is very hard to study mechanistically. And so this means that it's actually quite challenging to figure out, if you've got something that sort of works, it's very challenging to figure out how you can do medicinal chemistry to make it work better, because you don't know what effect it's actually having. So our idea was to look at this mechanistically, at what's going on in a single molecule. And we focused on this particular chaperone candidate. It's an iron tetrapyrrole that was discovered by John Collinge's group in the UK. And we chose it because it's actually been quite well characterized biophysically. So it inhibits prion replication in vitro. It also cures infected cells. They did isothermal titration calorimetry to show that it binds 1-to-1 in stoichiometry. And they even characterized the binding site from NMR. And most recently, a different group has shown that it has a property that's actually very important for antiprion compounds. Turns out there's different strains of prions, and some drugs that work on one strain don't work on another, which means you just knock down that strain and the disease switches to a different strain. So it doesn't help. So this one actually also acts on multiple strains. So we basically repeated our measurements, throwing in 50 micromolar of this tetrapyrrole. And when we did this, we observed 3 different populations of curves. So the first population, about 10% of the time, we saw curves that basically are the same as what we saw before. So they got the same unfolding force, roughly 10 piconewtons, and the same length. About 2/3 of the curves showed the same length change, but a higher force. So this suggests to us that the first one has no ligand bound, it's just the same as what we were looking at before. This one now has ligand bound to the native structure. And then about 1/4 of the curves showed no obvious rips, which tells us that this compound must have bound to the unfolded polypeptide and now is preventing the formation of the native structure, which is a little curious, and we'll come back to that. But there, whoops, sorry. Um, so the first thing we wanted to do is basically look at the effects of the ligand binding on the energy landscape that we'd characterized previously. So to do this, we need to look at the force distribution. So of course, we're only looking at the ones that do have an identifiable rip in them. And what we find here is that we have two peaks. So the first peak here matches actually the peak that we would expect for unbound PrP. This is what we got before. So this suggests that what we have is basically one of those cases that Olga was talking about yesterday. We have two different native states, effectively, one that's bound and one that's unbound. And so I need two peaks. So we just fit it to two distributions. And this then allows us to separate out the new distribution, the one that we care about, which is with the ligand bound. And then we can recover those landscape parameters. And when we do, we find that most dramatically, this distance to the barrier height, to the barrier has shrunk a lot, about a factor of 8 or so, 6 or 8. So it's much more compact and more brittle against force. The barrier has also gotten higher, but as it turns out, the whole complex is more stable, so it turns out that the barrier is roughly the same energy as it was before. Now we can understand this effect by looking at the structure. So if you look at where this thing binds, you find that it makes contact to the C-terminus here, and very close to the N-terminus there. So this is the N-terminus. So it's effectively acting like a staple across the protein. So it's not surprising that it makes it a lot more mechanically rigid. So we can measure the stabilization you get, and it's about 12 kT. And that matches what they found from the ITC before, which is good, so everything is self-consistent. So we can put together the bound to the red when there's protein bound. You can see it stabilizes. Image of how the landscape changes from the black when there's a lot, and it makes this much more brittle, so the barrier comes right over up to the folded state. So these results actually support the notion that Collinge's group originally put forward, that this thing acts just by stabilizing the native state. However, this left us with those quarter of the curves that were something quite different was going on, so we were curious, and we looked at these a little bit more carefully. And when you, say, take a bunch of these curves and you average them together to remove some of the underlying Brownian random noise, what we find is that it doesn't quite fit to a worm-like chain. Now, you might look at that and say, well, that's not super convincing, but that's what just a handle without any protein looks like. I mean, it's really quite flat. There's no residual. So this is real. It's small, but it's real, and it's consistent across all the molecules. So this suggests that to us that the protein is more compact than random coil. So there's some kinds of interactions that are compactifying it. But we see no observable rips, which presumably means that whatever structures are present, they must be fluctuating sufficiently rapidly that we're averaging over them in this measurement. And in fact, if we return to this picture of what the monomer can do, you might recall that these two misfolded states here, these were very rapidly fluctuating, and they were also not terribly stable, so they should only do things at low force, which is where we're seeing this behavior. So what we did is we took these numbers that we had gotten from our analysis of these two states in the previous data, and we plugged them into a model that for the extension as a function of force that we had developed for disordered proteins, where we saw similar rapid and marginally stable structures forming, and just predicted what we should see and overlaid it, and it actually matches matches great. So this is not a fit, right? It's just plugging in the numbers. So this suggests to us that what's happening is that this compound is binding to the unfolded protein, and it prevents the native structure from forming, and then that allows these quite unstable structures to remain for a little while. And so we pick them up in these measurements. Now, of course, you'd think this is kind of weird for something that's supposed to be preventing misfolding, that it seems to be actually enhancing it. But we were thinking that maybe this actually ends up being more important when we start looking at the interactions between different PrP molecules, because maybe it's inhibiting the formation of really stable aggregates, which would, in fact, be better rather than worse. And in fact, that's what we found. So if you recall, looking at the dimer without any compound, well, what we saw was typically things like this, where it forms this stable misfolded dimer. We never saw native folding. When we throw in the compound, you almost never see this. A lot of the time you see this, so it's like that shoulder feature that I showed you with the monomer. It's just twice as big. And in fact, if you put that same model and compare it to the average, you get something that matches it perfectly. But most interestingly, some of the time you see events that have 2 rips in them, each equal to the length of the monomer, which suggests that now we're actually able to form 2 natively folded domains. So what this suggests is how this compound is doing is it's actually inhibiting the interdomain interactions that stabilize the very stable misfolded states and gives room for the native structure then to form. So the picture we put together of the effects this is happening in the monomer without compound, you mostly get the native state. You sometimes get these little things, but very rarely, and they don't last. When you add the compound, the picture doesn't really change very much. You get a little bit more of these things. Okay. When you go to the dimer without the compound, you just see this misfolded state. You add in the compound, that inhibits the formation of this state, and instead you get these things happening. Now the curious thing about this actually is that some molecular chaperones from the cell actually act in a very similar way. They basically bind to unfolded or partially unfolded intermediates and suppress the misfolding through interdomain interaction. So this was some work done by Sander Tans in the Netherlands, looking at trigger factor and maltose binding protein tetramers, which tend to aggregate and form very stable things. And without the trigger factor, you see they just have a few intermediates, and then with the trigger factor, it stabilizes a whole lot of intermediates. And I didn't show it here, but it basically prevents the formation of these aggregates that you can't unfold. And it turns out that similar actions have been found with other chaperones studied at the single molecule level. So there's some very suggestive parallels here between the effects of molecular chaperones and pharmacological chaperones that no one has really explored. People have generally been looking at this in terms of just stabilizing the native state. All right. So with that, I'd like to finish the section on PrP and for the rest of the talk, discuss some much more recent work, which is as of yet unpublished, on superoxide dismutase. So this protein, this is a cytosolic antioxidant. It's natively a homodimeric beta barrel. Here's the crystal structure, and it is associated with ALS. So there's something like 140 mutations of it that have been identified with familial forms of the disease. It's only got 153 residues in it, so clearly it's very sensitive to mutation. And you find deposits of misfolded SOD. It's commonly seen in patients that even with sporadic, so non-familial, non-inherited ALS. And it's been shown to actually to undergo prion-like propagation of misfolding. So there's some connection there with the behavior of PrP. Now, normally this binds ions. They're required for its enzymatic function. If you look at the ion-bound form, the hollow form, it's actually very stable and aggregation resistant. But if you remove the ions, and especially if you also reduce the internal disulfide bond, it tends to form fibrils and misfold. And some people have suggested that maybe this species is toxic in vivo. Now, there's been a fair amount of study over the years on the folding of SOD1 using ensemble biochemical methods, and the consensus from this work is generally that you have a 3-step system here. So if it's a dimer, this first monomerizes, and then each monomer unfolds as a 2-state folder. Although there is some evidence that maybe it's a little bit more complex, but this is the general consensus. Now, the misfolding is much less well understood. Some people have suggested that intermediates could be very important, but there's not a huge amount of evidence for these intermediates up there. People have looked at transiently misfolded oligomers, and it's been suggested that the loops, there's 2 sort of partially structured loops loops 4 and 7, and these are thought to play an important role. But there's basically a lot of stuff unknown or not fully understood about how the misfolding works. So there's lots of questions to resolve. What is the misfolded structure? How does it misfold? What role, if any, do intermediates play, and how do the mutations actually fold into that? And then if there is an infectious structure, what is it and how does it propagate? So the approach we took is very similar to PrP. We start off with our molecule, either a dimer where we link the monomer domains together with a little peptide linker, or just a monomer, attach handles onto it in the same way as before, attach those to beads. We are studying the apoform, so there's no ions, and that's because it's the one that's most prone to misfolding. And there's 2 solvent-accessible cysteines which we mutate away. In fact, this is done in almost all the in vitro work because it makes it much easier to handle the proteins. Okay, so what do we see? Well, we first started looking at the dimer. And when we did one of these force extension curves, we saw 3 rips, which, okay, this seems to match the notion, right? We can check the length. The total length is 112 nanometers. When you go to the crystal structure, you expect for this construct, we should get 112.5, so that looks good. But when we measure the lengths of the intermediates, we find that this does not match what you should get from this picture of first monomerizing and then each monomer unfolding. That should give you a sequence of 4, and then 58, and then 112. And instead, our first rip is 50 nanometers, over 10 times bigger. So clearly something else is going on, and when this thing is monomerizing, the structure of the monomers are also getting disrupted at the same time. So the monomers must have some intermediates. Whoops, sorry. Okay, so because of this, we move to study the monomer first to understand what's going on there. So you can get an obligate monomer by mutating these residues, which disrupts the dimer interface. And when we do this and pull it, we clearly see something that is not two-state. Okay, now the total length here, 54 nanometers. In fact, if you do a couple thousand pulls, you get 54.7, which matches quite nicely with the expected length from the crystal structure. And we can fit these intermediates. There's obviously lots of them. Now, in fact, every time you pull on it, you see it's like you've got a bag of tokens, and you're pulling a few out, and you get some, and it's different every time you pull them out. So we see a variable number of intermediates between 1 and 5, never 0, so we never see it be two-state, but mostly 2 or 3 intermediates. And the intermediates you see from one pull to the next, they differ. So this is 3 different pulls from the same molecule, one with 1 intermediate, 2 intermediates, and 3 intermediates. So even though we are seeing different things each time, the same kinds of numbers repeat. And so if we make a histogram of all the numbers we see in all the pulls, we get something like this, and it's clearly got some peaks in it. And so we can fit the peaks in this distribution to identify where the lengths of these intermediates. And so we basically tile this distribution with peaks all of the same width, because that's determined by our instrumentation. And we find that we need, in this case, we have 8 peaks. And these 2 peaks end up being important because they're the last ones to unfold. Now this is highly suggestive to us because there's 8 strands. So we're thinking, okay, maybe what we're seeing is a peak per strand. Then we can look at the refolding, and you see exactly the same kind of behavior. Different numbers of intermediates, and different intermediates each time. That changes from pull to pull, but again, repeating numbers. And again, similar numbers of intermediates in each pull. And the distribution, in fact, ends up having peaks at the same locations. And in this case, these guys, where they formed last, I mean, they unfolded last, now they fold first. So we're basically visiting the same set of intermediates, doing both the unfolding and the refolding. So we can put an identity next to each of these. I'm calling them intermediates 1 through 8. And we can now make a transition map, visualize all of the pairwise transitions between states in all of the 2,200 curves. And this is what we get for the unfolding, where the width of the arrow indicates the fraction of the transitions that happened there. So you can see it's quite complicated. And then the refolding. So this is, what we see is basically a very large number of possible pathways through all these intermediates. And I didn't really mention it, but in the curves you often see sort of hopping back and forth a few times between states. So these transitions are fairly close to equilibrium. So they must have relatively small barriers. So this just suggests to us a picture where there's this complex energy surface with lots of fairly small barriers. And that's why we're getting so many different ways through it. Now, of course, we'd like to identify what these intermediates are structurally. And in order to do that, we basically enumerated all the possible combinations for unfolding each of the strands in the native structure. And then we basically enumerated the lengths associated with each of these and matched those against the lengths that we observed. So for example, this is one force extension curve that had 3 different intermediates at these lengths. And then we went through all this set of states where this shows you which strands are folded. So this has 1 through 6 folded, this has 5 through 8, this is just strands 1 and 2 folded. So these are all the, and we allow for some slop in the identification. And then we basically looked at it as a network and traced how can I get through from the folded to unfolded, because, for example, I can't unfold 1 and 2, so at 1, 2, 3, and 4, and end up with beta 5 through 8 here, and then get to any of these states, because I'd have to go backwards and refold. And so in this case, we end up with a few possibilities through these states. And then we do this for all of the 2000 over 2000 unfolding and 2000 refolding curves to see what matches what we actually see. And the picture that emerges is that the most likely structures from this for each of those intermediates is the following. So this first state here involves taking off the strand 8 and loop 7, which is attached to that. The next possibility is to remove strand 1. Strand 7 gives you the next state after that. And then, depending on which one you remove the other one, you get the next state after that. Remove strand 6, and then 5 and loop 4 at the same time, because loops, of course, can't withstand mechanical force. And you get over here. Now, there's another possibility that ends up being degenerate with this one in terms of the length, and that is that we remove strand 6 up here, and then we take the loop, so we change the order. And again, this state here is degenerated in length with that. We wouldn't be able to tell the difference. And this leaves us with then just 3 strands, then 2 strands, and then the unfolded state. And this accounts for, this sequence accounts for something like 85% of all of the curves that we see. So it's by far the most likely thing that's going on. So this suggests that each of those intermediates, in fact, corresponds to unfolding or folding of each beta strand sequentially. And those 2 states that I said were always last or first, those end up, we call this the stable core. It's these, this hairpin and then this sheet there. All right, now, about 5% of the time, we actually don't see that behavior. It does not end up with the right length for the native state. So we have intermediates that, partially folded intermediates that look the same as in the native curves, but the total length change doesn't go to 0. So it's shorter. The structure is smaller. So this tells us that it's a misfold. It's not the native structure, right? And these things last long enough that we can see them in these pulling curves. So on the order of seconds. And in fact, by looking at these lengths, we see that there's 3 different misfolded states. This one is the shortest. This one is closest to the native. And we can actually quantify what their lifetime is. It's about 6 seconds by looking at how these misfolded states decay by waiting longer and longer at 0 force for them to resolve themselves. Now, if we look at the intermediates that we observe on the curves that start or end in a misfolded state, we find that they share the same intermediates as the native structure, and they always have this stable core. So it's suggesting to us that when you fold up, you always form that stable core first, and then the misfolding happens around that. And this is consistent with the notion that from previous data that the loops are in fact what's prone to misfolding. Because we can identify all these intermediates, we can actually see where the misfolded state diverges from the native folding pathway. So what's the branching point? And we find that there's 4 different possibilities for the branching point. If we add those onto our map, it looks like this. And you can see that with the exception of this case, everything else is happening right before a loop is involved. All right. So armed with what we know now about the monomer, we're going back to look at the dimer. And for example, in this curve, right, the idea is that, okay, this strand 8 and loop 7, that's the weakest part of the monomer. So the idea is when we rip, and strand 8 is actually in the dimer interface. So when we rip apart the dimer interface, what we think is happening is that we're also pulling off strand 8 and loop 7 and possibly more of the protein. And in fact, this particular length would match up with something that looks like this as a possibility. So this is something that we are currently working on to try and analyze a whole bunch of unfolding curves and see if we can make sense of them in light of the monomer data. So with that, I guess I'm a little bit early. I will thank the army of people who worked on it. Team PrP, which is these guys right here. Team SOD. And our collaborators, so Val Sim and Leo Cortes, who in the Center for Prions and Protein Folding Disease. One of the problems with prions is that they require a biosafety level 2+ to 3, depending on what version you're working with. And so if we ever want to deal with truly infectious material, which we don't put in our trap, but we want to test things, so do some assays with them. So we have to go and work with these guys because they have a fully equipped biosafety 2+ facility. And then our collaborators on the SOD work, Steve Plotkin, the mysterious guy who I guess was supposed to be here, and Neil Cashman. And of course the funding agencies. And thank you.